Let's continue the secretory function of the gastrointestinal system with have a have a look at the stomach. You know that in the stomach we have several glands that are penetrating deep uh, in the mucous layer, and uh, in this anatomical structure, try to maintain the gland away from this high acid concentration that we have inside the lumen. If you look at the gastric glands, you can see that these gastric glands are composed of several cell types, and they form a way into the lumen that they are named as gastric pits. At the top layer and at the top of these pits, we have some surface of epithelium. They are named, as I told you, the gastric pits, and below them we have the mucous neck cells. The mucous neck cells, as the name imply, they are responsible for secretion of different mucus, the different level of mucus. That this mucus contain high level of bicarbonate. Therefore, protect these glands against the acid concentration and acid that is available just in the lumen. A layer or a little deeper area in the deeper area, we have parietal cells. These are the most important cell that we have in the stomach, and these parietal cells are responsible for secretion of acid chlorhydric. You know that the most well known secretion of the stomach is acid, and this acid is released by this parietal cell that you can see in this figure. The parietal cell also is responsible for releasing another hormone factor named as intrinsic factor that I will discuss about. That intrinsic factor is a factor that bind to vitamin B12 and protect vitamin B12 from the acid environment that we have in the stomach. Then this complex will be moved to the intestine, and inside the intestine the vitamin B12 will be absorbed. Without intrinsic factor, you know that B12 absorption will be impaired, and therefore we have a type of anemia. Another cell that we have a little deeper area, you see there are cells that are named as Schiff cells. You can see that the acid secretory cells are placed in upper part. Therefore, the acid content will flow immediately out of the gastric gland. Therefore, this acid that is dangerous will not be able to make any injury to the gland itself. In deeper area, we have the Schiff cell, and Schiff cells are responsible for releasing a very important hormone named as pepsinogen. You know that pepsinogen is a inactive form of pepsin. When the pepsinogen reach the gastric lumen, and in the presence of high acidity, this time pepsinogen will be converted into pepsin. In fact, I can say that pepsinogen is inactive form that will be activated in the presence of acid. It is important. The presence of acid is important. And the Schiff cell, therefore, is responsible for enzyme secretion. Except this cell that I told you we have in the gastric glands, there are some other cells that are very important for the function of the other cell of the gland, particularly enteroendocrine and cells that release hormones that are responsible for regulation of gastric lumen secretion. You know that endocrine cell that we have in the gastric gland release gastrin, uh, cholecystokinin, and these are responsible for regulatory mechanism that I told you before. There are also enterochromaffin cell likes that release histamine. That the histamine is a main regulator for acid secretion in the gastric lumen. This figure is showing a gastric gland in another view and another form in a schematic way. As you see, the gland is opening is closed. Therefore, the acid has no way to reach the deeper area. Instead, when acid is formed in the parietal cell, it will flow out of the gland into the lumen, and there is no way to come back. And you can see the peptic cell, or the cells responsible for releasing pepsinogen, that are located in deep area. And this is the muscularis mucosa, the layer that I told you we have been in the deep area of the mucus layer. There are some other layers that are working, such as the submucosal layer and smooth muscles, but here is just in the mucus layer. Now the question is that how acid, and this very, very strong acid, is being secreted by the gastric gland and the parietal cell. This figure showed a parietal cell structure. As you can see in the basolateral part, this parietal cell contain large amount of vessels and is in contact with large amount of vascular bed, therefore provide enough blood for them. And you know that blood provide carbon dioxide for these cells, nutrient, and so on. On the other hand, you can see that in the parietal cell, that it is also named as oxyntic cells, there are lots of mitochondria, and these mitochondria are responsible for generation of the energy for these cells. These mitochondria secrete their content these mitochondria provide ATP for the secretory function of the parietal cell. And you see that parietal cell have a way to secrete their content into the lumen. Now let me to review how acid is being formed. Firstly, carbon dioxide in the right figure, you can see that carbon dioxide is being absorbed from the interstitial space into the parietal cell. This is completely in reverse to other cells that the carbon dioxide will be absorbed by the blood from the cell. Now cell absorb carbon dioxide from the blood into the cell. This carbon dioxide then, under the function of one enzyme named as carbonic anhydrase, will be mixed with water, and then therefore carbon dioxide water will generate acid carbonic. This acid then will be dissociated into hydrogen ion and bicarbonate. Bicarbonate that is formed here will be reabsorbed in the exchange with color, and bicarbonate will be sent into the interstitial space and into the blood. On the other hand, hydrogen ion remain free here. This hydrogen ion then is able to be exchanged with potassium. One potassium will be absorbed into the cell, and instead one hydrogen ion will leave the cell into the lumen, and at the same time, I told you that bicarbonate is exchanged with color, and this increases the color concentration inside the cell. Then this color will move out from the cell into the lumen according to the electrochemical gradient, and therefore hydrogen and color will reach each other, and they form the acid coloritric. The exchange of potassium and sodium also help this process to complete it. Therefore, I can say that in one word, carbon dioxide from the blood is absorbed. It was converted into acid. Acid was dissociated. Hydrogen was secreted into the lumen and bicarbonate into the blood. And therefore, I can say that when the gastric cells are working, they are absorbing a net amount of bicarbonate. I can say that in an active gastric lumen, the venous blood that come out from the gastric lumen contain high level of bicarbonate. Therefore, they are alkali. And this is a process that is very, very simple in the formation of acid. Now, this acid secretion should be controlled. It is not easily secreted, but it should be controlled. How acid is being controlled? The main regulator for acid secretion is coming from the acetylcholine from the parasympathetic nervous system, gastrin from the G cell of the enteroendocrine cells, and histamine that is released from ECL cells. In this figure, you can see that gastrin, acetylcholine, and histamine all are able to increase and activate the increase calcium concentration and activate cyclic AMP to activate some protein kinases. And these protein kinases now are able to stimulate the potassium hydrogen pump. Remember, I told you that potassium is absorbed into the cell and hydrogen out of the cell. This pump is extremely important for us when they are activated, when these pumps are activated. And as you can see in this figure, the histamine, acetylcholine, and gastrin are able to stimulate this channel. Between these items, histamine is extremely important. For this reason, inhibiting or blocking the histamine receptor, H2 receptor, by antihistamine drugs, not those antihistamine that we use for allergic reaction, that this is another receptor, H1 receptor. But H2 receptor is responsible for the activation of the hydrogen potassium pump. Therefore, inhibition of this receptor can decrease the secretion of acid and the acid content of the stomach. This is the mechanism that histamine is involved in the regulation of acid. It shows that how much this histamine is important. On the other hand, you should know that even acetylcholine or gastrin that are able to stimulate the acid secretion directly, they also stimulate histamine secretion from ECL cells. And indirectly, they stimulate the acid secretion by stimulating histamine release. Therefore, I can say that regulators for the acid secretion are acetylcholine from the enteric nervous system and parasympathetic nervous system, gastrin from G cells, histamine from ECL cells, cholecystokinin from I cell, and secretin. These are a stimulant for acid secretion. And this figure shows what I told you before. Gastrin is released into the blood, and this gastrin either stimulate parietal cell directly or stimulate ECL cell to release histamine. The neurons, enteric neurons and parasympathetic neurons, release acetylcholine. Some of the acetylcholine stimulate parietal cell directly, but on the other hand, acetylcholine also stimulate enterocheiromuffin cell likes to release histamine. And therefore, I can say that histamine is the main regulator for the acid secretion. On the other hand, we have some inhibitory mechanism for control of acid secretion, particularly a negative feedback mechanism. Here you can see when inside the lumen hydrogen ions concentration is increasing, then it stimulates these cells to release a hormone named as a somatostatin. And somatostatin move and inhibit G cell and inhibit gastrin secretion. You can imagine what I'm going to say. I can say that a feedback mechanism from inside the lumen, when the acid content is increasing, immediately inhibit gastrin secretion, and therefore this inhibit the acid secretion. And this feedback mechanism regulate the concentration of acid in a limited area and limited value. This is a summary of all the items that I told you. Have a look. I explain you that vagal afferent flow stimulate that release acetylcholine, stimulate the parietal cell to release acid. Also, this vagal system stimulate the enterokeromorphin cell to release histamine. Also, this vagal system stimulate the G cell to release gastrin. This shows how much vagal system is strong, that is able to stimulate all aspects of acid secretion, not only just acid secretion. Also, you can see that acetylcholine stimulate Schiff cell to release pepsinogen. The enzyme release from the Schiff cell also is being controlled under the vagal system. And this figure show you one point. And what is this point? This point is here, that gastric secretion is very, very dependent on the activity of brain, activity of autonomic nervous system. And this shows us how cephalic phase of the control of GI system is working. I will discuss this and I will explain this in a little bit. This figure show a little more complicated figure, show that when I'm talking that histamine, somatostatin, acetylcholine, gastrin stimulate acid secretion, they involve tens of intercellular mediator, and these mediators eventually stimulate the acid secretion by the hydrogen potassium ATPase pumps. And by the way, let me to tell you that those drugs that inhibit the histamine action that I told you, such as ranitidine, inhibit the acid secretion. Sometimes we have some other drugs that inhibit acid secretion by directly inhibit the hydrogen potassium pump in this figure. This pump. If we inhibit this pump, acid secretion will be inhibited. And you know that drugs such as omeprazole is using this mechanism to inhibit hydrogen potassium pump. Therefore, acid secretion may be inhibited. Also, there is another way is to neutralize the acid that is already secreted. Therefore, we use some drugs to neutralize the acid that is secreted. Okay. Now the question is how the gastric secretion is being regulated. Just a little bit earlier, I told you about the role of brain in the regulation of gastric secretion and the neuronal mechanism that were involved in this way. Collectively, I can tell you that secretion of the stomach is being regulated in 3 steps, in 3 parts. One part is cephalic phase. Cephalic phase means the stimulant that come from the brain that affect the parasympathetic nervous system are able to stimulate this gastric secretion, acid secretion, enzyme secretion. And this phase is commonly take place even before food entry. Therefore, I can say that cephalic phase is a feed-forward mechanism. You remember I told you that in feed-forward mechanism, brain expect and predict that in coming minutes, coming seconds, food will come into the stomach. Therefore, before even the entry of the food, the secretion will be stimulated. This is a reflex mechanism. For example, when you are very hungry and suddenly the smell of a very delicious food come to you, your brain stimulates acid secretion even before you're eating your food. When you are in a restaurant, you are very hungry and you see the dishes on the table. You didn't eat the food yet, but just you see the dish, your brain stimulates the secretion. This is named as cephalic phase. But let me to tell you that the cephalic phase is not always stimulatory. Sometimes it is inhibitory. For example, when you are in a situation, for example, you are very distressed, or you are depressed, or you are very hungry, but you see a scene that is very, very bad for you, the food that you do not like it. These factors inhibit acid secretion, inhibit gastric secretion. Therefore, brain is able to either stimulate or inhibit the gastric secretion in advance. What about the second phase? I told you that there are three phases of gastric secretion, regulation of gastric secretion. What about the second phase? In the second phase, we have gastric phase. Gastric phase means when food enters the stomach, when food have contact with the stomach, when the stomach is being stretched by the food, distension of the stomach, stimulate the intrinsic mechanism that we have inside the stomach, such as enteric nervous system, such as hormonal mechanism. And at this situation, for example, the content of food, even when we have a food that contain high level of amino acid, protein, it stimulates acid secretion inside the gastric lumen. These are short reflex mechanism that we have inside the stomach, and this will stimulate the secretion of gastric lumen. What about the third phase, the last phase? It is named as intestinal phase. In the intestinal phase, when the partially digested food exit from the stomach into the lumen of the duodenum, the duodenum try to monitor the acidity of the food content, osmolarity of the food content, and accordingly send some signal from the duodenum back to the gastric lumen. For example, when acid content of the duodenum is increased and pH is decreased, the duodenum will inhibit acid secretion from the gastric lumen. In fact, the duodenum will tell gastric lumen that acid is more than enough. Please come down. Please release less acid. And this is the intestinal phase. Therefore, collectively, I can say that regulation of gastric secretion takes place in three phases: cephalic, gastric, and duodenum. This table shows different area, different region of the gastric lumen, and you can see the different stimulant. For example, in the oxyntic gland and in the antrum parts, the stimulus that when the acid is less than 3, pH is less than 3, it shows that there is very high acid environment, this will stimulate somatostatin, and somatostatin inhibit gastrin release and then a gastric secretion. This is a gastric phase and it is inhibitory. Have a look at the duodenum. In the duodenum, when acid is available or hyperosmotic solution are available, they release secretin and stimulate neuronal reflexes, and all this will and secretin inhibit the acid secretion by inhibiting gastrin. At the same time, secretin inhibit acid secretion. Nervous reflex that come from the duodenum inhibit acid secretion reflexly, and also hyperosmotic solution inhibit acid secretion, stimulate the duodenum and inhibit acid secretion. What about the duodenum and jejunum with regard of food content? As you can see, when we have fatty acid in the duodenum, high level of fatty acid, one enzyme that is released is named as GIP, and GIP inhibit not only acid secretion but also gastrin secretion. This is showing the different phase that I told you, gastric phase and the duodenal phase. Gastric secretion is not just limited to the enzymes and acid that I told you. The very important function of and secretory function of the gastric lumen is release of a mucosal protective layer that protect the mucosal layer from the high acid that we have in the inside the lumen. Have a look at this figure. I told you that inside the stomach we have very, very high acidity, that in highest acidity reach even the acidity of 2. The pH will reach 2. That it's extremely dangerous. But as you see, these cells, the mucous layer cells, release a very heavy amount of bicarbonate layer, that this bicarbonate form a mucous layer that is protecting against the acid content of the lumen. Have a look at this. The acid is here is 2, but when it reaches the surface of cell, it is 7. And this very large gap show that very clearly that this mucous layer protect the stomach content, stomach cells, against stomach content. And this is impossible. Without this layer, immediately the acid will make injury to the gastric lumen itself and cause the ulcer, different type of ulcer. Even in this in very bad situation, it can cause bleeding. And in normal situation, the healthiness of the gastric lumen is very dependent on the amount of this mucous layer, this protective layer that is released into the lumen. And different type of drugs, a stressful situation, or even sometimes smoking and alcohol drinking, all will undermine this mucous layer secretion. And you expect that those who are in very stressful situation, those who are using the drugs such as steroid hormone drugs, they are inhibiting this mucous layer, and this has nothing except that when the mucous layer is decreasing and is undermining, therefore the acid have a chance to reach the mucous layer cells and make injuries. That lots of people have this problem. They have peptic ulcer in their gastric lumen. And that nowadays we know that sometimes the presence of some microbiome also is involved in this process, such as E. coli, that make that participate in the peptic ulcer formation. This in these slides, slide, I told you about the secretory function of the stomach. In the stomach, digestion and absorption is very little. It is, I can say that in the stomach, just preliminary digestion of protein is taking place by pepsin. Some digestion of carbohydrate will be done. Because of the movement that I told you we have in the stomach, the grinding function of the stomach will take place. But we have very, very little absorption from the stomach because of this very thick layer that they have. Except those drugs that are very, very dissolvable in the, or they are very lipidophile, such as alcohol, such as some drugs such as aspirin, that may be absorbed. But generally we say that a stomach just release the acid and enzyme and mix it with the food, and preliminary digestion taking place, and then this content will be delivered to the duodenum. In the duodenum and then jejunum and the ileum, the absorption will take place. Okay, let the time of this, let finish this part and discuss about the secretion of the pancreatic secretion and other secretion that we have in the GI system in the next file. This figure, by the way, this figure show the effect of different factor on the rate of release of gastric juices. And as you can see, when we eat meal, immediately the pH is increasing because the food is mixing with the acid, therefore acidity is decreasing. And on the other hand, volume of stomach is increasing. Rate of acid secretion, as you can see in this dashed line, gradually increase to decrease the pH again and make the acid environment again strong. Okay, by this slide, let me to end this part. This is the part number 6, and I finished this part, and I will continue this part and this topic in the next file that I will send you separately. Thank you very much for now, and take care. Thank you.